Yes, dear student, welcome again after classification of virus, which we have discussed in my last episode. Now we are again and again, I said to you that virus is an obligate parasite. Obligate parasite means it require always host the parasitic nature of virus. Why it is parasite? We have also discussed that virus does not contain any cell machinery. It means that it hasn't got cell. It is not producing its energy. Whenever energy is not there, it cannot work. So, and the metabolic factor is also not there. The water is also absent there. And without water, no metabolic reaction can take place. So many other organelles are required. So many other things are required which are necessary for this functioning of life, which we call the vital functions. We require cell. So, non-functional, it remains non-functional outside the cell because there is no machinery. Only two things are there, proteins and genome, and it cannot work without any protoplasmic machinery. Now after that, it requires host for its structural molecule synthesis. Its structural molecule synthesis means what? For formation. I have told you it is made up of capsid, and capsids are made up of capsomer. And capsomers are made up of proteins. Protein units and proteins are synthesized by the ribosome. It means that, and as well as we have also discussed, some enzymes are also required, transcriptase and other. For the replication of DNA, if you want to increase its number, means in the life cycle, whenever it increases its number, so many viruses are produced by the host, what is required? Means number of the genome molecules are also required, and genomes are also synthesized by the process of the replication because it's making their copy. And whenever there is no enzymatic factory, there is no enzymatic things are there, enzymes are there, the DNA or RNA cannot be replicated, cannot make their copy. That's why. And after that, the proteins which are the structural unit or glycoproteins which makes the envelope of it. So, or some of the enzymes which are present in the viruses but remains inactive outside the cell. They are synthesized by the cell machinery, and cell is not present. That's why it is locating for a host cell, an active cell, which have all the machinery. So what is going on? That's why it requires host for structural molecular synthesis. Viruses has powerful genome. But the question arises here. All the living organism has their immune system. Whenever there is an immune system present in all the living organisms, how virus can enter into them? And how the living organism, the living cell, allowed to control them? It's very difficult, but the problem is that the viral genome is very strong and powerful genome. Whenever it attaches with any host, there are so many chances to overcome these genome into the, or overcome the genome of the host cell. That's why it can enter. Viruses has powerful genome to overtake and control the host metabolic machinery. Enzymes and organelles all has been, but the bacteria-like thing also has their own, just like the restriction enzyme. These are enzymes which destroy the viral genome as well. But it has a powerful system by which it can enter and overcome the machinery of the host. That's why it is parasitic. The virus has a parasitic nature and parasitic adaptation. With these, we have so many questions. Number of these students generally asking me in my class that, sir, when virus can die? Impressive question. Whenever I am saying that virus is a living thing, any living thing has to die. Now the question here, it is only, means the death of virus only takes place whenever its genome destroyed, means unable to synthesize the structure protein or its enzyme. If it is modified, it may synthesize the other type of the protein or maybe a mutant of that will be formed. So, in this way, this is the only way by which we can destroy them, or the nature can destroy the virus. Getting my point? Otherwise, it cannot be destroyed easily. Now the question arises, if it remains outside the host, it can survive for longer period. That's a longer period. Means so many years it is placed at a place and nothing will be happening with them. Why? The question arises. How viruses survive in the environment as well in the host? Whenever host has a machinery, then what happens? It can survive. It's a machinery, means that immune system is present. Whenever virus is coming in my body, my immune system will be activated against this virus. If my immune system will destroy the virus, what happens? I will be safe. But if it will overcome, just like the coronavirus, if the coronavirus will overcome the body, man will die. Getting my point? So many viral diseases can cause the death of the living organism. That's why it is called poison. So now the question arises, how viruses survive inside host? What happens whenever it enters into the host? It can destroy the host or degrade the how. It can degrade. What is going on? Whenever it enters into the host, it synthesizes endonuclease by, not its endonuclease. This endonuclease will be synthesized by the host to degrade the host DNA. Surprising. The enzyme does not belong to virus, but the instruction belongs to the virus. When it gets into that, just it controls the endonuclease system, produce endonuclease enzyme to destroy the host, and then it will survive in the host. Because the DNA become weak, degraded, and it become unable to control its own host cell. Then another way, deactivating the complement system. What is complement system? We have three lines of the defense. In immune system, we have three lines of the defense. The first line of defense is these surfaces, as well as some of the enzyme or the secretion which is present in mouth, nose, means this nose, ear, and other parts of the wherever the entrance is present in a mouth. Whenever the viruses and pathogen defeat this first line, it enters into the body. Then the second line of defense are there. And here, phagocytes, some cells are there, cytokines are there, and some complement proteins are there. These are also called plasma protein, which try to destroy the invader. Then what it do inside the body of the host? How it defeated the complement system of a host or complement protein. You know the mimicry, means making sound like that. This is called the mimicry. They are doing the same. What they do? They innate system, the complement system, innate means we have given by the God, by inheritance. The first line of defense, second line of defense is our innate immune system. So plasma proteins are the part of the innate system. And what they do? They produce mimic protein like complement protein. And the cell become confused. The mimic protein, its own protein, means its own site. They synthesized by the host, but it is not dangerous for the virus. That's why it produces mimic protein and cell become confused, which will be the friend, which are the friends of it and which are the enemies of it. So it survives. Block interferon response. Another type of the defense protein produced by the human being and others, these are called interferon. These are also the defense protein of the host against virus. So what they do? They block the interferons response or interferon synthesis. And whenever interferon synthesis is stopped or blocked, the virus survives. After that inactivation of the major histocompatibility complex system. What is this major histocompatibility system? Our each cell, each body cell has some markers, which is the recognition. Which is our recognition. My body cell, all cells have the same type of this markers by which my body identify these are my cells. Whenever someone comes inside my body, it has its own system of proteins, which is different from mine. It means that the body immediately recognizes these cells having my markers on it. These markers are different from my body markers. That's why they do not belong to me. That's why this is called antigen. The invaders are called antigen. And this is recognized by the helper T cells. Helper T cells is just like the Nadra of our country. The Nadra has all the data. If Nadra system will destroy, what happens? The data of all the peoples of the country and so many invaders from the different sides of the borders get into our country. The same is going to takes place here. What happens? Who recognize the helper T cell, recognize the major histocompatibility cells present. It is the virus come and destroy these helper T cells. Now what happens? The body is unable to recognize the invader, recognize the virus which will get into it, and the virus will enjoy in the body because no one can recognize he is or it is the invader. Getting my point? Next, after that, the another cells, lymphocyte and B means B cell, you can say the white BCs. The white BCs are of different types. The helper T cells are the type, but the two lymphocytes, the B lymphocyte and T lymphocyte, but the lymphocytes are the type of the white BCs which mature in my bone marrow are called B cell, and which mature in my lymphatic system. Lymph nodes are called T lymphocyte. So B lymphocyte and T lymphocyte, but the B cells produce antibodies. These are the proteins as well as. These are the antibodies which bind the antigen, means the invader. Antigens are the invader. Antibodies are the defense proteins. These defense proteins block the invader. These are called antibodies. And what they do? The blocking proteins are not going to be produced because they are suppressing the B cell activation. B cell remain inactive. They are not producing their antibodies. So who will block the virus? So virus will enjoy in my body. So this is one way that how virus survive inside the host. Now, environment, which is very much severe. High temperature, ultraviolet light, the acidic, basic condition, so many things, dryness, humidity less, humidity high, so many harsh conditions in the environment are present, or maybe very favorable. If favorable, virus will enjoy. But if it is not favorable environment for the virus, what the virus do? The virus will try to get a shelter somewhere. And in this way, it will survive. It will do something with that. So if you will give a special condition, then it will destroy, otherwise it will not destroy in the environment. Now let's come to that. Let's come to see that how the virus survives harsh condition of the environment. The first virus survival in temperature. What they do? They get shelter in some of the dung, water and other things. Organic molecules, they get inside it and the temperature effect will not occur on it. And in this way, the virus will survive. Not by themselves, but something remains inside that one. So, DNA has more endurance, means its DNA can bear the harsh condition because it is present inside a shelter. The next, airborne survival in humidity. Now what happens? Humidity is important factor whenever airborne something like. So whatever it has a special covering of the humidity. It has a special covering of the lipoprotein. It has a special covering of the other things. So what happens? They make a covering. Until this covering will remain there, they will survive. Like that, lipids envelopes are more weak. Means if they have lipid envelope. What is going on? Means especially the envelope virus are weak viruses. They can destroy easily. And if it is envelope viruses, so non-envelope, they can survive. Because the lipoproteins or the glycolipids can destroy easily. Therefore, the airborne survival, virus survival in the light, especially some viruses which are in connection with the phytoplanktons. Phytoplanktons are unicellular algae. These are called phytoplankton. Light come on this unicellular algae. What they will do? They will photosynthesize and produce food. Produce food means it is producing energy, capturing energy. And whenever it is capturing energy, it will survive, phytoplankton will survive, and virus get its component from there, and virus will also survive. If the light will be dim, or light will not be there, then what happens? Then it is difficult for the virus to survive ultraviolet light. Ultraviolet light destroy virus as well as the other phytoplanktons. They are the same. Here, humidity. The humidity, if humidity will high, they will survive. Especially the enveloped viruses, survival rate in less humidity is 20 to 30%. But non-enveloped virus has 70% survival rate. After that, here pH factor. The pH, the ideal pH, is near to the neutral one. The neutral pH is the 7. And 6.5 is the ideal pH for the viral survival, though not the nature of the protein, not denaturation of their lipo, means denature or destruction of their glycolipids and lipoproteins which are present. It's the ideal condition. But if it will below to the 6.5 means 4.5 or something like that, or maybe high as 7.5 or 8.5, what is going on? The lipoprotein will destroy. And whenever the lipoprotein will destroy, then there is a chance of that. If it is enveloped, it is easy, but the lipoprotein also dissolve in the organic matter. That's why we use sanitizer to destroy the virus. We use phenolics in our houses, means phenyl and other compounds to destroy the virus. We use Dettol soap, we use carbolic soaps, different like Lifebuoy, new Lifebuoy, to destroy different type of the Kataru. These are the things. So these are the pH. Lipids hydrolyze in basic pH and organic solvent can destroy. So if it will remain away from these type of the things, low and high pH, if it remains away from the phenolics in other, if it will get shelter. If the humidity is around them is wonderful, then it will be survive in environment. Otherwise, there might be a problem for the viruses. Thank you very much. Next time, inshallah, we will discuss the bacteriophage and its life cycle. The structure of bacteriophage as one of the specimen of the virus, and its life cycle means how viruses produce their new ones, or as an example we will use